In this video we will discuss about some basic idea about transfection and uh, an overview what actually the transfection means. So what is transfection? Transfection is a method to insert foreign nucleic acid into eukaryotes. So what does this mean in simple terms? In simple terms, it means that it is used to alter or modify the genomic sequence that is present within the host. So in order to do so, there are several factors and there are different kind of transfection based on what kind of final outcome we want. uh, so let us understand initially how it happens. This is the transfection agent that helps the nucleic acid sequence, that is this, to penetrate inside the host cell. So how this happens is, first this transfection agent will cover this nucleic sequence, and this nucleic sequence is susceptible or easy to penetrate within the host cells, or any penetrate the plasma membrane of the cells. And then when it is inside the host cell, in the cytosol of the host cell, this transfection agent uh, will release this nucleic acid sequence. And then as we know that from DNA to RNA, if it is a DNA sequence, it will convert it into RNA and then protein synthesis. So this process of the uh, translation of the foreign nucleic acid sequence can be either done in the cytosol or it can be done in the nucleus. It depends on the kind of nucleic sequence that we had. Uh, During ER stress, we have understood about NLS, that is nucleation uh, sequence. It means that this nucleic acid will find the final outcome of this nucleic acid sequence will be in the nucleus, and there it will transcribe and then translate. However, in this, both the options are shown. One is the DNA has been converted into RNA and then into protein synthesis in the cytosol, and another one is DNA converted goes to the nucleus, and there the regular translation transfection process occurs, and then protein synthesis come outward. Another major factors, one of the major factor, or we can say one of the major factor in transfection is the transfection agent. That defines the efficiency of the transfection. If transfection agent is not good, then transfection efficiency might not be good, and the whole experiment might not be good for your functional studies. So in order to do so, the various approaches has been identified and various modification has been done on those approach. One of this is the cationic polymer. So at the time of transfection, people use cationic polymer to increase the transfection efficiency. So some of the couple of uh, cationic polymer are polybrine or hexadimethyl bromide. So these are used along with the transfection agent that contains the nucleic acid sequence or the plasmid DNA, and that finally helps in the increased transfection efficiency. And here in this graph, here in this figure, we can see that the cationic polymer and specialized modified cationic polymer has been made, that coupled with transfection enhancer. So this in overall will enhance the transfection efficiency, or we can say enhanced cellular uptake. Cells are more uh, prone to uptake this kind of configuration. So this paper has the paper one of the paper has work on this kind of enhancer, and here we can see that if the cellular uptake increase, it will then go to the usual process that we have already discussed in the previous figure, that it will go to the nuclear transport and then gene will express. So one of the example of this uh, is to give is to insert a fluorescence sequence, or so that any modified cells can be observed uh, with the help of fluorescence, and this will enhance the data measurement compared to the other methods, which are mostly or sometimes indirectly. So this is considered as one of the uh, great approaches, or great findings or inventions that has enhanced the data interpretation, and at the same time more accurate data can be gathered from the biological experiments and with more reliability. So this is just a overview about transfection and how it is useful in nowadays biological experiments. That's all for this video. Thank you for your time.